Coronavirus control treatment of common colds is symptomatic, no vaccines or specific drugs are available. Hygiene measures reduce the rate of transmission. Introduction: coronaviruses are found in avian and mammalian species. They resemble each other in morphology and chemical structure. For example, the coronaviruses of humans and cattle are antigenically related. There is no evidence, however, that human coronaviruses can be transmitted by animals. In animals, various coronaviruses invade many different tissues and cause a variety of diseases, but in humans they are only proved to cause mild upper respiratory infections, i.e. common colds. On rare occasions, gastrointestinal coronavirus infection has been associated with outbreaks of diarrhea in children, but these enteric viruses are not well characterized and are not discussed in this chapter. Clinical manifestations: coronaviruses invade the respiratory tract via the nose. After an incubation period of about 3 days, they cause the symptoms of a common cold, including nasal obstruction, sneezing, runny nose, and occasionally cough. Figs. 60:1 and 60:2. The disease resolves in a few days, during which virus is shed in nasal secretions. There is some evidence that the respiratory coronaviruses can cause disease of the lower airways, but it is unlikely that this is due to direct invasion. Other manifestations of disease such as multiple sclerosis have been attributed to these viruses, but the evidence is not clear-cut. Figure 60:1. Clinical manifestations and pathogenesis of coronavirus infections. Figure 60:1. Clinical manifestations and pathogenesis of coronavirus infections. Figure 60:2. Immunopathogenesis of coronavirus infections. Figure 60:2. Immunopathogenesis of coronavirus infections. Structure: coronavirus virions are spherical to pleomorphic enveloped particles. Fig. 60:3. The envelope is studded with projecting glycoproteins, and surrounds a core consisting of matrix protein enclosed within which is a single strand of positive sense RNA, Mr. 6x106, associated with nucleoprotein. The envelope glycoproteins are responsible for attachment to the host cell and also carry the main antigenic epitopes, particularly the epitopes recognized by neutralizing antibodies. OC43 also possesses a hemagglutinin. Figure 60:3. Electron micrograph showing human coronavirus 229E. Figure 60:3. Electron micrograph showing human coronavirus 229E. Bar, 100 mn, courtesy S. Sicotra, Leicester Royal Infirmary, Leicester, England. Classification and antigenic types: the coronaviruses were originally grouped into the family Coronaviridae on the basis of the crown or halo-like appearance given by the glycoprotein-studded envelope on electron microscopy. This classification has since been confirmed by unique features of the chemistry and replication of these viruses. Most human coronaviruses fall into one of two groups, 229E-like and OC43-like. These differ in both antigenic determinants and culturing requirements. 229E-like coronaviruses can usually be isolated in human embryonic fibroblast cultures. OC43-like viruses can be isolated, or adapted to growth, in suckling mouse brain. There is little antigenic cross-reaction between these two types. They cause independent epidemics of indistinguishable disease. Made by Mohammed Nadeem.